Okay, so now we can move on to the most important part of the day, the nano part. It's my honor to invite to present Dr. Joseph DeSimone. He is the inventor of print technology. He's going to talk about this. This is a top-down method to create nanoparticles. He has over, I believe, over 140 patents, 80 pending patents, over 300 publications, very renowned expert in the field of nanomedicine.
Thank you very much for the invitation to participate with you today in celebrating the new facility and everything that's going on here in New York at Mount Sinai. I thought what I would do would give you a little bit of context of where I'm coming from and the kind of work that we're doing, and start with that we have one of the Centers for Cancer Nanotechnology Excellence funded by the National Cancer Institute. And like a lot of the work that was described here earlier today, this is a bridge between the clinical and the basic and physical sciences and engineering. And we try to bring all that together in a very translational way to help patients. The way our center is particularly organized is that the nanotechnology work that we do comes in both therapeutics and imaging, but it's typically focused on new imaging modalities. And so we sort of span the basic to the translational. We have projects focused on nanoparticle-based chemotherapy and biological delivery, but we also have carbon nanotube-based new imaging modalities. And given the nature of this audience and some of the things you're doing, I thought I would just spend a moment and just walk you through what my colleagues are doing in that regard. And then I'll end on the nanoparticle therapies, which is what my group's focused on. We too are celebrating a new building. Our building's not quite open yet, so we're drafting behind Mount Sinai. Our building opens up in April, but it's the largest building on our campus. And on the first floor is our imaging facility. So this is an imaging research building. It's got a suite of MR machines, an MR PET machine, just like you all have here, will be in the central bay, and then an MR CT. And on the second floor, I'm very excited about this building. It's dedicated to nanomedicine. So it's an entire floor focused on nanomedicine, and that bridges the rest of main campus in the physical sciences and applied sciences with the clinical side here. But as I mentioned to you, there's some real exciting work that my colleague Otto Zhao is leading in the physics department. And it's a carbon nanotube technology, and it's an array of carbon nanotubes on a surface. So it's not a therapeutic, but it actually acts as a cold cathode associated with generating X-rays. And it has a really fast rise time, so fast, in fact, that one doesn't have to swing an X-ray source around a patient. You just have to turn on these switches. And you can have multiple sources of X-rays using this approach. And this is actually a clinical machine that's actually already in our hospital that's doing breast tomosynthesis. And so it's actually under clinical trials already at UNC hospitals. So it's a fundamental new way of delivering X-rays. Coupled with that is some new therapeutics or new therapy approaches in developing a microbeam radiation therapy system off of this same basic technology. So it's got a lot of imaging focus, pun intended, but also is moving towards a therapeutic approach here with the radiation therapy. It's pretty exciting. Coupled with that program, we also have, like you all have here, a really heavily focused capability in small animal imaging tied to mouse models. And we have great capabilities on it. And this is a core facility in our Cancer Nanotech Center. So that's just sort of an overview of how we sort of structure things at University of North Carolina and how we're organized. Let me pivot to what my work and the students in my group are focused on. So a little bit of context. I'm a material scientist by training, and have been doing only medically related work for about 10 years. And so I have that sort of flavor of material science in our background. And so when we look at a cancer cell, we're intrigued by things that not everybody finds interesting, but cancer cells are 10 times more mechanically deformable than regular cells. That deformability facilitates their distribution in the body and getting into places that are normally not found. And metastatic cancer cells are on average 10 times more deformable than regular cancer cells. And in fact, deformability is a phenotype of cancer. And we find that interesting. The mechanics play such a huge role in that disease. Bacteria come in a wide range of sizes and shapes. And I love this quote here. To be brutally honest, few people care that bacteria have different shapes, which is a shame because the bacteria seem to care very much. And it's interesting that the shapes of bacteria have been the result, obviously, of a lot of environmental selective pressures for their survival and function. And that these pressures have given rise to shapes and sizes that facilitate, for example, access to nutrients. Diffusion is very important on this length scale, so surface to volume ratio plays a big role. Motility plays a big role. And so 90% of the bacteria that are motile are rod-like to facilitate their translation. And those bacteria that are found in really viscous environments are not only rod-like, but they're helical to facilitate their translation, much like a corkscrew going through a cork. And so we find these kinds of properties, mechanics, sizes, shapes, all to play a really interesting role. And if you look more carefully at mechanics, there's a huge range of mechanical properties in biological systems, ranging from bone to very soft tissue that covers many decades in scale. And in fact, when you look at different cell types, and I already mentioned to you that cancer cells are almost 10 times more deformable than regular cells. But when you start looking at other types of cell types like red blood cells, which are mechanically very deformable when they're young, they get stiff when they get older and facilitates their removal. Or if you begin looking at pathogens, and a lot of pathogens that trigger infectious diseases or trigger cancer, they have significantly wide-ranging mechanics as well. So my group is interested in properties of biological systems like that. And the one that I think I'll just end on, just one more example, is in infectious diseases. When a phagocyte, macrophage, dendritic cell, approaches a pathogen, that there is an immense amount of information, it's now known, that's conferred at the earliest onset of phagocytosis. That when there's at the synaptic interface, so to speak, when the pathogen is interfacing with a phagocyte, the functional groups around the pathogen, the size of the pathogen, the mechanics of the pathogen, all start to confer information at that onset of phagocytosis. And that information starts to tailor the inflammatory responses and the immune responses. And so there's a lot of information that the immune system has evolved around to take advantage and understand these sizes, the shapes, and the mechanics, and the chemical distribution. So we find all that very interesting. And so we have a simple hypothesis then, is, can we take some of that information in biological systems, size, shape, mechanics, chemistry, and apply that to making better medicines and better vaccines? That's what we want to do. So, okay, if you want to do that, then you sort of are faced with, okay, well, how are you going to fabricate things with a kind of exquisite control that would be necessary to start to mimic these kinds of properties? And to me, this then becomes a real material science-like program. Obviously, you have to interface with biology and the life sciences and ultimately clinicians. But we start with what we're really good at, and that's on the material science side of the house. And so, one thing you could sort of argue is that a lot of the biological effects that are influenced by these properties may not be revealed by traditional particles, because traditional particles often don't have that kind of control of sizes and shapes, or they're very polydisperse in size and shape, but also chemically heterogeneous. And so given that, what my group has focused on then is our entirely new strategies for particle fabrication. And in particular, we have focused in on the microelectronics industry and the techniques used to make transistors in the semiconductor industry. We have adopted and completely modified for our own needs so that we can make organic particles that can mimic some of those properties I just showed you and give us exquisite control so that we can actually start doing clinically relevant things in medicine and infectious diseases. So we're bridging two very different fields, microelectronics and life sciences. And for the young people out there, I know we have some high school students. I think it's really important to think about, where are you going to study? What are you, how are you going to make a difference? And I think strategy is often all about being different. And one of the most ripe places for innovation is often at the interface between two very disparate fields. And so that's what we're trying to accomplish by trying to bridge microelectronics and medicine using this approach. Okay? And this is not to imply we're going to make babies in a clean room. So nobody should, that's not a reverse arrow. That's a one-way direction there. How many people have heard of Moore's Law? So anybody want to render a definition of Moore's Law? Yes, you had your hand up still.
Every 2 years, the piece of technology will be half size and twice as powerful.
Yeah, and there's a lot of variants of that. I think one would be the number of transistors would double every 18 months. Some people would say the clock speed would double, but who was Moore? And he founded a little company called Intel, right? So, and so this law, this observation has been going for a long time. And in fact, if you look at these techniques to fabricate transistors, the transistor hasn't changed very much in the last 40 years. They look basically the same if you didn't look at the scale bar, if you didn't look how small they are. And in fact, when you go back to the early 70s, the chips were still about the same size. They may have been a square centimeter, a computer chip, but on that square centimeter, they could only fit about 2,000 transistors on that square centimeter, because the size of a transistor, the smallest size of a transistor back then, was about the size of a single cell, about 10 microns. Today, you buy a chip, and you buy the 62-core Xenon chip, they've been able to cram 5 billion transistors into that same relative area, and now the minimum feature size is about the size of a virus particle. So Moore's Law, in biological terms, over the last 40 years, has managed to go from the size of a single cell down to the size of a virus particle, these fabrication techniques. And in the last decade, they've been in a size regime that's close to the sizes found in micelles and liposomes and some of the and viruses and some of the traditional and non-traditional delivery vehicles found in nanomedicine. And so this is what we're trying to do, is can we harness these techniques now that they're in the size range to make nanoparticles where we have the control of the microelectronics industry. So we developed a technique based on this we refer to as PRINT, particle replication and non-wetting templates. And again, back to the young people out there, if you come up with an acronym that's got some stickiness to it, like this one, it's powerful for your group and your career and brand building. Okay? So people refer to PRINT. And in the beginning, my group, we initially called this non-wetting imprint lithography. And the acronym for that was NO-WILL. And you didn't want to be branded with a terrible acronym like NO-WILL. So we came up with PRINT, and it works much better. So basically what we do is we lithographically define silicon wafers. We developed some liquid polymers that are essentially perfluorinated. These were perfluoropolyethers. So think about polyethylene glycol. Replace all the hydrogens with fluorine. Now you have a liquid that is a liquid down to minus 120 degrees. It's got an exceedingly low surface energy. It's got a positive spreading coefficient, meaning it will wet these surfaces. And we decorated this polymer with functional groups such that we can shine a light on it after it wets this surface, and it solidifies into a mold. Using light in just 2 or 3 seconds, it will solidify, and we can peel it off, and it's basically a Teflon-like mold or ice cube tray with cavities that are defined by the lithographically defined master template. Okay? And so what we've done now is we've converted this to a roll-to-roll process. We make that mold, and then using techniques from the old film industry, we could spread liquids on this mold and do a film split technique with another film that has a much higher surface energy, and we can uniquely fill the cavities without wetting the land area between the cavities. So it's a simple molding process, okay? Once we solidify that liquid, we can then bring that in contact with another film that has an adhesive, pulls all the particles out of the Teflon-like mold, then we can simply dissolve away the adhesive, release the particles, and begin the process in the formulation. And so there's a lot of unit operations associated with this that I won't get into, but the fundamental one is a continuous process and allows us to mold particles of uniform size and shape and uniform chemistry. And so this is a collage of some of the particles that we fabricate out of different chemistries and different length scales. And obviously we've got shape control. And you can start to see these filaments. These filaments are only 80 nanometers in diameter. And we can vary the length. We can vary how stiff they are. Okay, whether you think about my Italian heritage in me, you like your pasta al dente or well cooked, you can vary the stiffness of these hydrogels. And what role does that play on the biodistribution when you start injecting these by different means? We can also start controlling where the chemistry is within particles, and we can do spatially controlled approaches to maybe even help cells communicate more effectively. B cells and T cells and other things that we can start facilitating those kinds of approaches. We've spun out a company called Liquidia, and this is a film right from Liquidia. This is now a GMP compliant process, so good manufacturing practice that allows us to mold particles in a way that allows us to do translational science and put products into people. And you can see the sort of colors, the fluorescence or opalescence associated with the diffraction and playing with light, and it's a really robust process. My academic laboratory has a machine very similar to this. It's not GMP compliant, but we use the same molds that Liquidia uses in their GMP compliant process. So it's actually a very seamless translation. When we scale up a new design, they can easily translate it to their machine, and it facilitates the translational aspects of it. So what we do is we think of this as a platform, and we begin thinking about, now that we can make things and we can scale them up, we can start thinking about what applications we want to start pursuing. And I want to sort of give you some examples of this. I'm not going to be able to cover all of this, but I'll focus a little bit on inhalation, a little bit on vaccines, some on oncology, but we've designed synthetic blood and red blood cells that mimic the real red blood cells in mechanics, contain hemoglobin almost at the same amounts. We have some real exciting work in ophthalmology, and are moving multiple products into the clinic in that field, and so a lot of different things. But I'll just give you a flavor of this. And I'll cover three sort of general topics. One is I'll talk about engineering endosomal escape. How can we take unique chemistries in this dry method of particle manufacturing and applying it to designing particles that can degrade inside cells and release a cargo? How can we take advantage of particle size and shape and mechanics on circulation times? And then I'll try to pull some of this together in some of the applications that we're pursuing. Let me start with inhalation. My colleague wrote this article here, 1,000 Years of Pharmaceutical Aerosols. Clearly that was before the FDA. And I think some of these aerosols were recreational drugs way back when, and maybe even today. But this is what dry powders typically look like. There's been no engineering control on particle size in a simple thing like a dry powder aerosol. And size and shape plays a really big role in aerosol characteristics. The regional deposition in the lungs, where do they deposit in the lungs? How do they couple to primary and secondary airflows? Once they deposit, how do they get through the mucosal lining? How do macrophages interface with these particles? The ability of delivering macromolecules. As big as the biotech industry is, there is no inhaled monoclonal antibody on the market today. A lot of the big pharma would like to be able to have delivery of biologics via dry powders. And so the opportunities are really enormous. And clearly size and shape plays a big role in aerosol and flight characteristics. We all know that. We can just look at helicopter maple seeds. And as they fall from the tree, these are auto-rotating objects in low velocity air streams that actually create a leading edge vortex, create lift to facilitate their dispersal. So we can now start to take those kinds of characteristics and design particles with off-center masses so that they have a tendency to rotate. And when they start to rotate, they can actually change their sedimentation rates. And we can design particles and do computational fluid dynamics and actually make particles that change very different how quickly they will settle and how different they will couple to different airflows in someone's airway. Not only their flight characteristics, but their dispersal characteristics. This is pollen. In fact, this is a North Carolina pollen. It should be the state bird of North Carolina. We have a lot of this, pine pollen, and we design particles similar to that. And so we have powders that if you just have it as a quiescent pile of powder in your hand, it takes very little energy to aerosolize these powders into an aerosol, and it does it at really high efficiencies. Most aerosols, you think about for asthma and COPD and pulmonary hypertension, not much gets aerosolized. It's only like tens of percents. And using particle design, we can get into 80, 90% of aerosols that can be dispersed and use the drugs very efficiently. The particle can be 100% of an active ingredient. This is 100% cinamivir molded using print. 100% of an enzyme. We can mold pure biologics. These are 100% siRNA, molded as a dry powder. And just like a cube of sugar next to your cup of coffee in the morning, as soon as these drop into a wet environment, they quickly dissolve. Right? So these are just held together in a dry powder formation, protected from humidity in a blister pack, and can be dispersed directly as a dry powder. And so there's a lot of opportunity. Our company's got a big partnership with GSK. GSK is one of the leading providers of dry powder inhaler formulations. And just to give you a sense of the kinds of things we can do, and again, I'm not going to go into these details, I just want you to have a flavor of it. These are powders with really narrow size distributions. And most particle technologies, you cannot tune such a narrow size distribution. And what's important about that, especially for aerosol delivery, if the particles are too big, when you inhale them, they can't turn into the lung and you end up swallowing them if they're too big. If they're too small, less than a micron or so, you inhale them and then you'll exhale them just like smoke. And so there's a narrow size range where you want to get regional distribution and deposition. And we quantify that. We end up using imaging modalities, and so we'll label these particles. We'll begin looking at deposition. This is work in a canine model, technetium-labeled particles. And we can begin, as we vary the particle size, from larger particles, they tend to be in the upper airway. And then when we get into smaller particles, we get deep into the alveolar regions of the lung. And so we can quantify differences in different animal models with different particle sizes and shapes, how well they deposit in the airway. We're very interested in particles that would have a very low inflammatory response in the lung, so we could do extended release of drugs. We're very interested in treating lung diseases. We're even interested in preventing lung disease and lung cancer for people that have been smoking much of their lives. And so a chemopreventative approach using this. But I'm going to skip some of that and just show you the other kinds of things we're interested in. So one of the things you sort of ask is, well, what role does shape play on phagocytosis and macrophage uptake and clearance? This is alveolar macrophage. And so now that we have the ability to control size and shape, our laboratory and a few other labs, Samir Mitagatri, have been trying to quantify what role does size and shape play on uptake. And there's some really interesting learnings that we're having relative to particle size or lobe size of the particle relative to a cell surface ruffle, and some of the ease with which particles can be internalized by phagocytosis. And so we begin looking at a range of different particle sizes and shapes, and we get into mechanisms of frustrated phagocytosis and shapes that macrophages really love, and we have other shapes that macrophages don't like as well. And so we can start to tailor, if you want a drug, if your site of deposition in the lung is the macrophage, maybe for tuberculosis, we'd recommend a certain shape. If you don't want your drug to be eliminated prematurely by macrophages, we'd recommend a different shape. And so we can start testing hypotheses based on the location of the mechanism of action. This is a fun video. Cell taking up our particles. We actually catch this cell dividing, and one of our particles is spanning two daughter cells and causes the cells to refuse. And it ends up being an unstable cell with two nuclei, and that ends up dying. It's a fun video to watch. But we're doing a lot of observational science now and more detailed analysis of where particles go, what are the rates of uptake. And we have a whole set of particles now that the macrophages clear very quickly, and we have other shapes that they don't, and we're now testing in vivo hypotheses with that. But you know, once a particle enters a cell, and these are ovarian cancer cells now, once particles get internalized by different mechanisms, and I'll get into that more later, what can we do to facilitate release of a cargo? And can we begin designing, you know, Trojan horse approaches for cargo release? And if you look at cell uptake, you know, these are some particles that have a fluorescent dye on them. Let's see if we can get this. And that's pH sensitive. And the particles are blue externally, and once they get inside the cell, you'll see them turn red. And that's a quick drop in pH. And so we've been quantifying the pH drop as a function of particle size and shape and endosomal size. So you know, you get some of these unusually shaped particles, and you ask a quick question, what's the shape of the endosome that has a particle that looks like a hex nut in it? Is it spherical and the hex nut sort of floating around in that endosome? Well, it looks like the endosome is actually a shrink-wrapped endosome. It's toroidal. So that shape has a very high surface to volume ratio. And the proton pumps that lower the pH of the endosome now has a lot more surface area, and it looks like the kinetics of pH drop is much faster as you get into these high surface area endosomes. And can we take advantage of those characteristics to facilitate endosomal escape? And so we've designed some chemistries that do just that. These are hydrogels where the cross-link is a bis-silyl ether. And you read that in this paper here. As you vary the substituents on silicon from dimethyl to diethyl to diisopropyl, you can change the rates of hydrolysis by 5 orders of magnitude. So the particle is a network. It's a hydrogel. So technically that means it's a single molecule because it's one molecule. As the particles get internalized, we put a green dye on particles made with this cross-linker. We put a red dye on particles made with this cross-linker. These hydrolyze quickly, these don't. And you can start to see that the green ones swell pretty quickly, and they eventually dissolve. As they dissolve, they're creating molecules. The osmotic pressure, the particles are caught in an endosome, they start to fall apart, molecules are released, the colligative property of osmotic pressure goes up, water floods the endosome, ruptures the endosome, and dumps the contents into the cytosol. And it becomes a mechanism for engineering endosomal escape. And we can watch this by electron microscopy because we know what the shapes were, non-degraders and degraders, and really begin engineering this. And so what you can do is you can trap chemotherapeutics into these hydrogels. Those particles on cells that contain chemo, they get internalized, they degrade, and they release the chemotherapeutic into the cytosol. So it becomes a really potent way. Again, I love this video. It's kind of like carpet bombing. You can just watch these cells just die very quickly. And so we try to put all these strategies together to design better therapeutics that only degrade once they're inside the cell. So let me just, again, I'm going a little slow here. Let me pick up the pace. We have a big oncology effort. We do a lot with different small molecules. We have a really strong pharmacokinetics core. We do all the maximum tolerated dose studies. We look at the distribution of drugs and particles, and we bring that all together in efficacy models. One example is with Taxotere. We heard about that this morning. Taxotere is used in a lot of different cancers. We've been able to formulate Taxotere as a solid-state solution in PLGA particles made by print. And because of the dry nature of print, we're able to access a drug loading that far exceeds what's in the literature. And we think it's because of the way we make these particles, we're able to access that. And so we can make particles with a wide range of drug loadings, but the same size and the same shape. And then we end up looking at where's the drug, where are the particles in different tumor models, and then pull that together in efficacy studies. And so we can start asking questions about, what role does size and shape play on the pharmacokinetics of a drug? And you can start to see, much higher plasma levels for certain shapes versus the free drug, Taxotere. What's typically delivered in the clinic is a small molecule formulation. And then you take that same drug and you put it in a particle, and you see significant changes in the pharmacokinetics. We end up having a really significant increase in area under the curve in the plasma exposures, and we can have a significant Cmax increase. But then you begin looking at the clearance rates. We have really high clearance rates when the drug is delivered as a free drug versus in the particle. And you have dramatically lowered clearance rates, and the volumes of distribution are really significantly much lower. And what this is derived from, and a lot of people in the audience know this, is a classic EPR effect. When you have a small molecule drug and you give it by an IV, it can go everywhere. And therefore, the volume of distribution is really, really high. If you take that little drug and you put it in a massive thing called a nanoparticle, which seems like a wrong word for something like that, but it's a big thing relative to the small molecule, that big thing keeps it in circulation. It keeps it in a circulatory system. And therefore, the volume of distribution is much, much lower. Okay? But around a lot of tumors, it's actually fortuitous that there's a lot of gaps due to angiogenesis that are on the order of 200 nanometers or so. Too small for a red blood cell to get through, but small enough for a nanoparticle to get through. And therefore, we end up having selective deposition of a drug in a tumor kept in circulation for a long time to facilitate tumor treatments. And so we can put all that together, and we're plowing through a wide range of different animal models now with known drugs like docetaxel. Here's a non-small cell lung cancer model comparing Taxotere versus the nanoparticle approaches. We have a significantly higher maximum tolerated dose, or MTD, with the same drug delivered in a PLGA particle than as a free drug. And again, I'm not going to go through all these details, but we've published this in a number of different areas. And we're taking standard drugs, formulating them in a PLGA, and using them going forward. One thing I will say, it looks like the smallest dimension plays a really important role in distribution. And these are some of our smallest particles that we make now. And I put this up because of our discussions. These are some 55 nanometer particles that would be interested to combine with some of the things that you all do. I'm going to skip. We do some chemically modified forms of docetaxel. We lipidate it. That changes its rates of hydrolysis. I'm going to skip that. These turn out to be pretty effective as well. I'm going to move to hydrogels and get into mechanics. These are hydrogels made out of polyethylene glycol-based systems where we vary the cross-link density and therefore dramatically change the mechanics. And we can make them easily out of a wide range of sizes and shapes, as well as how easily they're deformed. And then we also pegylate these. And so we're trying to optimize the best particle, size, shape, mechanics, and pegylation strategies. And one of the ways that we often screen for circulation times is we do intravital microscopy. So what we typically do is we'll do a tail vein injection of particles that are fluorophore labeled. And then we'll use an intravital microscope, and we'll just put it on the earlobe of a mouse and look at the arteries, the veins in the ear of the mouse, and use that to track our particles. And so I'm going to show you some particles here that are not well pegylated. So they're cleared very quickly, and we're going to put a red dye in those. And we're going to co-inject those with a different particle that's highly pegylated, and we're going to put a blue dye in those. But we're going to co-administer them, and we're going to watch them on different channels in the intravital microscopy. So let me show you that. So the blood vessels light up because the nanoparticles are throughout the blood very quickly, and then you can see that it's essentially cleared, but these are the macrophages that have gobbled up the particles. They've turned bright red. Over here, I'll play this again, this stays blue, and you don't see the macrophage clearance nearly as long, nearly as significantly. And you can really see it in the overlay here. I'll show that one more time. And you can see it in the overlay that the red particles are selectively cleared and the blue ones are not because they're highly pegylated. And so it dramatically changes elimination half-lives. And you can pull all that together and calculate the elimination half-lives. And what we find is with the highly pegylated particles, we end up having an elimination half-life of about 20 hours for these particles of this particular size. These are 80 nanometer by 320 nanometer cylinders. So we're systematically going through this in a range of different systems. We attach drugs to these hydrogels using this bis-silyl ether as a new pro-drug linker strategy. And again, you can read about this in JAX. And this allows us to control the release of drugs very efficiently. So this is a combination of combining particle size, shape, mechanics, and the kinetics of distribution of the particle, and then we overlay on those kinetics particle drug release rates that we can tune with this strategy to optimize an ideal nanomedicine. And that's how we're trying to go about doing this. The drugs that we can link are not only small molecule drugs, but they're biologics. We have a really exciting program with siRNA delivery. Again, it's taking advantage of, in this case, a reducing environment inside the cell. So in this case, we want to fix a cell, not kill a cell, or sometimes we want to kill it too with siRNAs approaches. But there's a really efficient way to deliver siRNAs using this approach. You can layer on targeting strategies onto these particles. And that's, so there's a cascade of complexity that we're adding on as we need to improve therapeutic response. And so just to give an example, these are some particles that we've conjugated Herceptin onto the particles, targeting breast cancer receptors. This is a cell line without that receptor. This is a cell line with the receptor. And you can see significant uptake once we start doing these kind of targeting strategies. And why we think that's so exciting is that unlike traditional antibody drug conjugates, where you may only have 4 drug molecules per antibody, if you go to a nanoparticle, you can have on the order of 10 to the 5th chemomolecules per antibody, because most of the particle is drug. And that allows us to do these kinds of targeted strategies with other drugs. So let me end on vaccines. Clearly, vaccines plays a huge role in infectious diseases as well as cancer. You all know about the human papillomavirus and Gardasil. This is a quote from Bill Gates. In the same way that during my Microsoft career I talked about the magic of software, I now spend my time talking about the magic of vaccines. I had a chance to meet Bill about 3 years ago. And on that sort of quick, I knew I was going to meet him, and what do you say to Bill Gates to get his attention? And it's the elevator speech, almost like the poster blitz we did today. And I thought long and hard, what was I going to say to Bill that would get his attention? And I got introduced to him. He said, Bill, Joe DeSimone. Joe, this is Bill. I said, I know who this is. And Bill says, what do you do? I said, Bill, we're using the manufacturing tools of the computer industry, Microsoft, to make new vaccines, the Bill and Melinda Gates Foundation. And he did sort of this WTF look, and it turned into a 30-minute conversation. And I'm excited to say that our company, Liquidia, got the first equity investment ever by the Bill and Melinda Gates Foundation in a for-profit biotech. And it's because we're now heavily working on vaccines. And I already told you that pathogens come in a wide ranges of sizes and shapes and mechanics and surface chemistry. Okay? This is what Bill is dedicating his life to, and Melinda as well. And when you look at vaccines, first generation vaccines were live attenuated whole pathogens, right? Give the pathogen, chemically deactivate it, heat treat it, inject the pathogen into somebody, your body recognizes this pathogen, and you have a really robust immune response. But there are safety issues, especially if you're immunocompromised, elderly, children, and so people don't like live attenuated whole pathogens as vaccines. So what did everyone start pivoting to? Well, take the pathogen and basically grind it up chemically. Strip off some of the viral coat, maybe strip off a polysaccharide off the flagella of a pneumococcus bacterium, and just give those molecules. And the pattern recognition receptors in our dendritic cells and macrophages all recognize these molecules, and you'll start to elicit an immune response. But remember what I just told you, you lost a lot of information about the pathogen by giving these small molecule fragments of the pathogen. That synaptic interface is no longer there. And so what we're trying to do is recapitulate the original pathogen by combining the molecule approach that are all the classic vaccines today and put those back onto particles to get back the information that was lost when you chemically grind these up. And so what I love about this, it's this interface of material science and immunology trying to understand the proper design. And so again, I'm just going to quickly go through. I know we started about 10 minutes late. We do imaging just like in cancer. You do a subcutaneous injection of your particles, or intramuscular. Where do the particles go? Right? So this is foot pad injections. We watch the particles traffic to the popliteal lymph nodes. Right? We can pull the lymph nodes, and we can start looking and interrogating the lymph nodes and see our nanoparticles within the lymph nodes. We can pull the cells out of the lymph nodes and begin doing flow cytometry and look at which cells take up our particles, macrophages, B cells, D cells, as a function of particle size, shape, and zeta potential. All these characteristics play a really big role. And so, reinforcing the importance of well-characterized nanoparticles on zeta potential, size, and shape. We begin looking at IL-1 beta generation, so we start to see if we're triggering the inflammasome, working with the innate immunity group at UNC, and PEG hydrogel particles and PLGA particles made by print do not activate the inflammasome. There's no IL-1 beta generated. I will tell you, if you take a PLGA particle made by another technique and it's rough on the surface, it has a different response. And so when people say this material is not triggering the inflammasome, you can't say that. You got to say more about that. Is it particle rough or is it smooth? That stuff plays an important role. And so it's not just the chemistry, but it's the size, the shape, and the surface, and all those things come together. So we begin adding adjuvants, and we start adding different molecules that are agonists to different toll-like receptors and nod-like receptors, and then we pull all this together for our vaccine program. And so we have a vaccine program in ovalbumin, malaria, big, real exciting program in dengue, working on a bunch of other different vaccines. And I'll just give you a flavor of this, some imaging. 80 nanometer by 180 nanometer cylinders. This is accumulation in lymph nodes from 24 hours to 72 hours after foot pad injections. This is all at 72 hours as we vary size. These are all negatively charged from 80 nanometers to a micron. So the smaller particles traffic to the lymph nodes better than the big particles. And the negatively charged particles traffic better than the positively charged particles. You can start looking very carefully at the lymph nodes and begin seeing which cells the particles are in. The B cells are blue, the dendritic cells are green, the particles are red, and so the particles that are co-located with dendritic cells are yellow, which is what we want. And so we get 10 to 15% of dendritic cells in the lymph nodes taking up our particles, which is really high when designed properly. And so you put all this together and you can start designing vaccines. And this is actually a particle that we put into the clinic last year. So print is GMP compliant. These are PLGA particles designed with DC cholesterol. They're positively charged. You take those, you take the same influenza vaccine that you and I get every season, a trivalent flu vaccine. You cannulate this into the serum bottles. Within a few minutes, 90% of these molecules absorb on the surface of these, and we get a 12-fold improvement in immune response just by presenting the antigen more efficiently on the surface. And this is all the paperwork that you have to submit to the FDA to move into the clinic. So we've now tested this on about 180 elderly in Florida, and now we're beginning to move into next generations of these products. The last one I'll leave you with is this vaccine. When Lipitor went off patent, the biggest pharmaceutical product now is Prevnar 13. It's a vaccine against pneumococcus bacteria. Every child in the developed world gets it. It's a 4-dose sequence. I see some young mothers maybe nodding their head. It's a 4-dose sequence. They charge $90 a dose. The cost of goods is $22 a dose. It's Prevnar 13 stands for 13 different strains of pneumococcus bacteria. And they have to co-deliver 13 polysaccharides. These molecules have a molar mass from 500,000 to 3 million grams per mole. And you tell a bunch of chemists you need to co-deliver 13 molecules plus a protein. And what do chemists do? They link them together, right? But the linkage chemistry has a yield of about 15%. There's 13 molecules. 0.15 times 0.15 times 0.15 times 0.15. It's a very low yield, expensive process. Well, we can co-deliver these on our print particles with very high efficiency. We co-deliver them, but we don't covalently link them. We call this a virtual conjugate. And you look at the response. This is a commercial product Prevnar, 4-dose sequence on the prime. This is the antibody titer to this one polysaccharide. There's no response yet. You do a booster shot, you start to see performance. You now do this with a print particle, and all the animals respond on the prime. It looks like it's 70 times more efficient, and it looks like we're going to be able to do it as a single dose for maybe under $1 a dose. So this opens up global access. It also changes the financial dynamic in the developed world. And this is why Bill Gates invested in this company. It's pulling, I'm a firm believer that technology can drive costs down. So we have a pretty exciting program making new versions of really difficult to make vaccines and trying to epitope or engineer epitope presentation really efficiently. We're pretty excited about where this is going. But it builds off this platform, and each one of these programs feeds on each other, whether it's adjuvant delivery or chemotherapeutic delivery, targeting strategies and the like. And it's it's really integrated in that regard. It takes a village. My team is a team of chemists of all different flavors, material scientists, pharmaceutical scientists, biomedical engineers, MD, PhD students, all coming together to pull a program like this off. And this is our company, Liquidia. And as they say, vision without resources is truly a hallucination. And the opportunity to do what we do is a really great group of sponsors for our program, which we're really grateful for. So with that, let me end. I'd be happy to answer a question or two. Thanks. Yep.
That was really great. My one question is, you didn't mention the blood-brain barrier. Do you have strategies that might get targeting to go across that barrier?
Well, Genentech has that new transferrin targeting system that attenuates the affinity. We're looking at those kinds of approaches, but we actually see, and I don't want to come out yet, because we see much higher brain locations of drugs with our nanoparticle systems than a free drug. It's not, the particle is not crossing a blood-brain barrier, but I think it's it's contact diffusion is where I think it is, leading to these systems. So I don't know the mechanism, but we're seeing some real exciting data in that regard. There was a question up front.
Great presentation. So,
Wait for the microphone.
oh, sure.
You do it in stereo.
All right, great presentation. I wanted to know, is there some type of plan for the nanoparticles which aren't uptaken in the enhanced permeability regions? And when you're putting a chemo drug in there, which is toxic to the body, is there some type of plan for that to go through once the nanoparticle breaks down? What happens to the drug?
Yeah, no, it's a great question. I mean, our strategy is that we end up dosing maybe 15 times less drug than you normally would, but you're delivering it more effectively. And then maybe those scenarios would allow you to not worry so much because you have that with a small molecule already. And now we reduce it dramatically. But I say all that, we're seeing significantly higher maximum tolerated doses, MTDs, for a nanoparticle version of some of the common drugs you're very familiar with. And so how all that translates into new dosing sequences and area under the curve, I think all that has to be factored in. But we're seeing really significant efficacy changes, but we're dosing at some of the higher levels, but we're getting them on target much more effectively. So it's a complex answer. I think it's hard to make general statements. But it'd be really cool to have the particle nicely cleared if it doesn't get where you want to get. And I think there's some cool strategies that you may come up with to do things like that.
Thank you.
Yeah.
One quick question, it looks like you are using passive targeting approach, you are targeting the stuff based on the size of your particle. So have you considered toxicity issue especially for inhalation or intravenously if you are injecting the particle, it goes to the circulation, but there is possibility that it's not targeting to the specific cell, so you'll get some kind of toxicity.
Well, I think it's a little bit similar to the previous question in that, that's why we choose PLGA, to start with, we're choosing some of the same drugs that are general poisons. But again, as another layer of protection, as you start getting to specific kinase inhibitors, there's ways of putting specific drugs in there that might not be as detrimental, but now they're delivered more locally. It would be another layer of protection that one could add to that. Yep.
So you looked at inhalation and vaccination via injectable. Have you looked at other methods, for example, oral vaccines?
We're doing inhalation vaccines. Yeah, we're also doing sub-Q versions. The print array of particles, if you don't look carefully at the scale bar, and if they were really pointy, you'd see a microneedle array. And we just had a paper coming out in microneedle versions of these. The microneedles are for local delivery of chemo and inflammatory breast cancer model, but it's also being used as a way of removing cytokines that are triggering inflammation and scarring victims. And so we've got a big, so we're looking at multiple delivery mechanisms: transdermal, inhalation, sub-Q, intramuscular, IV. Oral, we don't see a whole lot of value yet. I say that, and my students will change my mind, but in size and shape control, and we haven't done much in oral. There's probably a lot of opportunity there, but we haven't started.
So, one last question. One? Oh, you want to? Two. I also have a question.
So you want to pick it?
I'm first.
We'll do two. I mean, like,
So, I can use
Yeah.
this microphone, right? Okay. So I'm interested to get a little bit of a sense of how shape control works on smaller sizes.
Yeah.
And so I saw you could go down to maybe 20 nanometers, that kind of range, but that obviously starts affecting the ability to make these funky shapes, right?
Yeah.
So maybe you can comment on that.
Yeah, so the question about shape on smaller systems, it's not quite clear yet. I think it may be interesting, some of the subtle details of caveolin mediated, where some of the gaps and the pits are much smaller, that might be playing a role. But it's not quite clear yet where all the opportunities are. I think at the macrophage level and some of these micron-sized particles for inhalation, it's very obvious for us. For tumor penetration, I think the minimum size coupled to deformability so it can extravasate through some of these tumors are going to be playing an important role. Where size begins to break down to be less interesting, it's not quite clear to me
Yeah,
yet.
but I'm talking
Or shape,
about
shape.
The technological challenge,
Oh.
right? Because at one point you come to the size of the polymer itself.
Oh, yeah.
Right? And
Well, we're
then-
getting close to that.
Yeah, so that's
We're getting close to that. I mean, we're down, if you would have asked me 10 years ago,
Can you get a donut that's 20 nanometers?
we've molded my cells and replicated my
Okay.
cells. So we've molded carbon nanotubes, right? So which are 2.5 nanometers in diameter, right? We think we have a resolution of 0.4 nanometers.
Okay.
And we have a paper published in Small on that. So the technology will go further, but for scale up, we're limited by what we make a lithographic pattern on. So we're drafting 1 or 2 generations behind Moore's Law. Their computer chips at 22 nanometers, we're at 55 right now.
Okay.
And it's just a matter of dollars, commitment of dollars for us to scale it up.
So you need another conversation with Bill Gates.
Yeah, but the question is, does it matter or not? And I don't know how far it's going to matter. I don't know.
so maybe to take a little bit on this, and I just read actually this week, something coming in from Microsoft, talking about going and doing transistors all the way down to the atom
Yeah.
size, right? And so
Or spintronics.
right, can this technology be adapted as well?
Well, we've molded down to 2.5 nanometers. So the problem is, when you get that small, you really have to want these particles. So I mean, imagine how it's kind of stupid to think about making things in a two-dimensional way. I mean, if you think about it, you're not going to fill a tank car up with that. You got to really want these particles, and they got to bring value. Against a three-dimensional reactor, you have a lot of advantages. All right, so nobody's going to be making paint using this approach. So you got to think about that value-added applications, and clearly in vaccines and some of these places we're hitting it. But there's a point of scale. So right now, these 80 nanometer particles, we make 0.5 milligram a linear foot. We'll run at 20 feet a minute, and we make what we need. And that machine is not that expensive. And this will support, we think we can get through all the vaccine markets, all the oncology markets at these smaller sizes, certainly all the ophthalmology markets. I mean, so there's places where this fits and there's other places where it might not.
Thank you.
Yeah.